Okay, I will try to consider mainly the subject here. It will have strong relationship to the previous lecture, and the relationship have some similarity to the next two pictures. This is a picture that Ben sent us once, but somebody else from his family sent this picture. And just to see it again. So there could be different view on the relationship between fluctuations and energies. Both of them are complementary, and I will try to describe what we learn about molecular motors. So the main part of the talk will be how complex molecular machine or motors work. And the same approach that will be used here is and could be used to other large macromolecular systems like ion channels. Maybe I will say something about it. And even for more complex systems like the way the ribosome insert protein in a translocon. But nevertheless, most of the talk will be on how we try to model molecular motors and to basically emphasize what is the question. We want to understand these systems based on physical principle. And in the same way that when you look on this system and you took physics, you will be able to calculate the work that was done by this water by knowing the weight of water and how much height was spent by this side and this side. So we want to try to get similar pictures and in some way see the similarity between molecular motors to regular machines. Now, since we talk on fluctuation, there is, it refused to work. This was supposed to show you something. It was working before, maybe we'll see another one later. But what it's supposed to show you is that this rod is fluctuating up and down. And whenever it is down, the driver pushes foot down and gets stuck. So it gets stuck here, the rod goes up, it stays here, the rod goes down. Again, you push the foot on the brake, and you overall climb uphill. Let's try one more time. Okay. So the question to the smartest young people here is, originally I thought that this will never happen, but it does happen. So why it happened? Is it because of the fluctuations of the rod or something else? Okay, so with this in mind, we go back to the biological systems. We have proteins. We now also have the structure of these molecular motors. And we want to convert the structure to function. And in most cases, I would argue, you need computer modeling to do it. So the approach that one would take, depend on the size of the system, is what is called now multi-scale modeling. You try to build your model, but always having solid physics, at least on the molecular level. Now, one way of this multi-scale modeling is the way you study, or you could study the chemistry in the molecular motor, which in this case is ATP hydrolysis. And the generic way to do it is by this so-called combined quantum mechanical molecular mechanics, which would take the part where you break the bonds and describe it quantum mechanically, and the rest of the system by molecular mechanics, while considering mainly the electrostatic effect of the surrounding. Now, this type of approach have been around for a long time, and now when quantum mechanical programs become more efficient, you could use it to answer really complex questions about, for example, phosphate hydrolysis, questions that were not resolved experimentally. This is trying to study GTP or ATP hydrolysis in solution by high-level quantum mechanics. And when you do this calculation, you could at least study what happened in solution. And one of the questions that you could resolve is a recent controversy whether the reaction involved a direct proton transfer from the attacking nucleophilic water or it goes through another water. And in very recent years, it appears that this mechanism is more likely, at least in water. And the way you summarize your finding are always by this potential surface, which takes huge amount of work. What you see here is the first step of the associative attack of the oxygen on the phosphate. And then you could ask what happened in the proton transport case. And if you want to study it, we wrote this huge review, which was chaperoned by Bent in Quarterly Review, which answered different questions. Now, after you study the chemistry in water, then you could move to the biological system, where it's still not reliable enough to do the same type of free energy calculation. So what you do, you use an approach which is called the empirical valence bond, train several state systems to reproduce what you know about the quantum chemistry in solution, and move with this type of simpler surface from water to the protein. And this approach, which I will not talk about, allow you to do very long-time simulations and also to produce nice movies of the hydrolysis reaction. And most importantly, you could use this type of approach to calculate the free energy for different mechanisms and to find how, for example, the ATPs accelerate the reaction. Now, this part is not what I'm going to talk about, but it's something that you have to learn to do when you go to the overall action of the ATPs. Now, the main question I will try to ask is how to model the whole action of this large molecule, where you have to overcome two issues. It's a very large system. If you will do microscopic calculations, they are unlikely, at least at present, to converge. And you also want to do very long-time simulations. So we have strategies for both of them, modeling large systems and doing long-time simulation. The first issue is simplifying the energy surface of this molecular motor. And the approach goes back to something we did with Mike Leavitt in 1975. You replace the side chain by spheres, which is an approach that is used now quite frequently in folding study. But in order to study the function, you have to teach the model electrostatics, which is, as you will see, the basic of most of the action. So you have to teach the model about correct interaction between charges, to teach it about the solvation energy, the energies that you pay moving charges from water to the protein. It ends up with relatively simple formulas which are very useful to calculate absolute folding energy, and essentially this model was calibrated by trying to fit the absolute stability of different proteins. And more recently, it was also calibrated on properties of membrane proteins. Now, there will always be the question of how reliable is this model, so it's always good to have an answer. And the way we assess the reliability of the model or answer hard questions is that we use an approach that we like to call paradynamics. That is, we calculate the free energy of moving from one point to another by the simple model, but then we do perturbation from the simple to the full atom model at the initial and final point. So we have a way to go to the reality of atomic systems. It does not mean that we use it all the time. Now, the second issue is how we simulate long-time processes, millisecond and even longer. So this brings us to a dilemma which changes with time. Do you want to use specialized machine or very powerful supercomputers, which are advancing more and more, or at present do you want to use these simplified CG models in Langevin dynamics? Both approaches are valid, except that at present, to the questions that I will be asking, you get faster answer by doing Brownian dynamics and simple surface. And here one could ask, how do you know that you have the right friction to study these molecules? And for this, we develop an approach that we call renormalization, which go like this. Instead of running the very large molecule and looking how it moves from open to close, we apply very strong forces on the atomistic molecule. With strong forces, it will respond in short time, and apply the same forces to the reduced model. The reduced model has a friction and Langevin dynamics equation of motion. So you play with the friction until both the large molecule with large force and the small molecule, the small system with friction behave in a similar way. It's basically what some electrician does when he checks electric system. You apply different voltages and try to figure out how the system behaves. This type of approach could be used to look on single molecule experiment and so on. And let's assume for a minute that it will allow us to study the dynamics. With this in mind, we go to the key questions of this lecture. How ATPase works, what is the source of the conversion of chemical energy to mechanical energy. Again. So we have this central stalk, which we will try to see how it rotates. We have chemistry here, and we want to understand how in this case, ATP to ADP rotate the stalk, or the opposite, when you have this stuff rotating, how it does ATP synthesis. Now, okay, this is the system. There is lower part which use pH gradient to rotate this stalk, and there is upper part which does the chemistry. We already heard in the previous lecture about these nice experiments, fantastic experiment essentially that show the rotation. You also saw this rotation, and we try to understand what does it mean. Now, one of the most exciting findings of this experiment of Kinoshita and Noji was that when this central system rotates, instead of going in jump of 120 degrees, it go 80 degrees, stop, and then continue. One of the questions is why this happened. Second, is it general? And the third one, is it really needed by evolution? And we will try to answer all of them. So the first question is, could you reproduce from the structure this motion of 80 degrees where you stop at what is called the catalytic dwell when you do the chemistry, and then you continue? Now, there are countless explanations of how this system works. A lot of them are spherical model, elastic spring model, even entropy. None of this model could actually describe the energetics that will support this 80-degree stalling or any other feature. So we use our simplified model, which is mainly based on electrostatics. And the first finding was that this model reproduces a pathway or landscape where it's stuck at 80 degrees because of electrostatic feature of the system. And then it continues in this direction. And my first reaction was, okay, we understand how ATPase works. It must go in this direction. As you will see in a minute, I did not fully think about it, though this statement is correct. This is the feature in two coordinates, the motion of the subunits that do the chemistry and rotation of the central stalk. You have to add to it the chemistry of the ATPase, and then you end up with this relatively complicated landscape where here is the rotation. At 80 degrees, you cannot rotate anymore, so you do chemistry where the barrier is around 12 kcal per mole, and then you go to this point where you release the inorganic phosphate and you are downhill by around 10 kilocalories per mole. Now, when we publish it, I felt that this explains how ATPase exert torque, because obviously you go downhill from here to here, and I felt that this leads to the torque. In a minute, you will see there was one element missing. Now, if you draw the same stuff in the chemistry and rotation of the stalk, you get this type of strange landscapes that we will discuss shortly to see if this specific pathway is really important. A footnote, we use our renormalization and we were able to reproduce the fluctuations. By the way, the fluctuations are random fluctuations. But as you will see, they need the energy to know where to go. So we could reproduce the time and everything, but then we are back to the question. Could we explain the torque that the single molecule people are measuring from this landscape? So again, we know how to do it in high school physics. We want to do it from our landscape. So when we try to ask the question, could we reproduce the torque here? And as I told you, I felt that just this 10 kilocalorie downhill of moving from ATP to ADP should explain it. We realize that if you will release 10 kilocalorie per mole at 0 degree and also at 120 degrees, you will have no torque at all. So if you have a system where you just do chemical work, but it's the same work before and after the torsion of the central stack, you will have nothing. So you need this type of tilted surface, which is in fact what we calculated from the first time, but we did not ask the question. So what you want to do is to take the landscape where you have both the chemistry and the conformation, and to calculate the free energy associated with rotation along this coordinate. If you calculate it and you get around 10 kilocalories per mole, it means that all the ATP energy was converted to torque. If you have a landscape that does not do it, it means that you don't have an efficient machine. So here we go to interesting works that the Japanese did. Because of the question, what really leads to this rotation, they done a series of amazing experiments, slowly removing all of this central part. And what they found is that it is still rotating. That does not matter what you do up to this point, it's still rotating, which means that we do not have that all the steric model or mechanical model will have difficulty to work because there is very little here to respond to the change of the subunits. If you look, however, on this electrostatic signature, you find that before and after the mutation, you still have this direction. And what we've done recently, it's mainly published in this new QRB, what is the name of this journal? Discovery, whenever it will go out. In the native, we do have all the torque. If we mutate the central stalk to all glycine, this disappears. So we feel that we reproduce the torque and we understand where it is coming from. Of course, there is more to work, but we've done one more thing. We tried to see if this mysterious 80 degree is important. And it's very easy to do it because you could decide at which place you do the chemistry. And what we found that actually the 80 degree catalytic dwell is not essential, at least not for this model and probably not for evolution. So of course the question is why it is there. And if, as a footnote, there are some ATPRs where it does not happen at 80 degrees. So I will have a few more things to say, but could anybody tell me why this car is going uphill, bent? Oh. Yes, so it's because of the driver, but it's actually because he have a good knee and he's pushing it down with ATP. So still there is energy here. And the fluctuations are important. They what guarantee the Boltzmann, but you need driving force. I will show you another case where this is apparent. And again, because of the previous talk, we have excellent introduction. This is myosin 5, which walk, and it does walk in one direction. So people who saw it, basically what is very common for a lot of systems is that we walk equally to both directions. What biology managed to do is this. So there is this asymmetry, and the question is, and this is a question that a lot of single molecule do not ask, why it goes in one direction and not the other? Essentially, a lot of single molecules succeed to stop the unidirectional motion, but there is the question why it is going in one direction. And there are basically two possible answers. One is that there is free energy gradient moving in one direction. The other that it is more kinetic barrier, that one of them is higher. Now, in order to resolve it in a satisfactory way, you have to spend a lot of time reading. I mean, it's very, very complicated. There are many steps. You have to basically learn about them, estimate the height of the barriers, and then try to ask, knowing the kinetics, what it is about moving in one direction. And this must be done looking at the structure. So if you look at the structure, the only source for asymmetry that we could find was in this connection, where it appears that this structure has lower energy than this one. Now, if in this type of 11 kilocalorie energy difference is, again, very hard to obtain by full atomistic simulation, but it obtained quite easily with our simplified model. And now, if you combine this information to the experimentally observed barrier and do all the accounting correctly, because it's really issue of accounting, you find that going forward has lower barrier than going backward. And you could see it maybe better with this diagram. When you move here, your backhand is bent when you do the main change in structure. If you try to move backward, you have to use the straight one. Now, after we wrote it, people say, well, we don't see what is happening. So one of the ways to see that this is actually correct is to try to do the kinetics, I will not call it dynamics, of moving like this and like this. And you could do it by Langevin dynamics. So this is using the model with Langevin dynamics and more or less the relevant barrier, and it does move in one direction. And one of the things we are trying to show now that if we apply opposing force, what type of force will stop the unidirectional motion? How we are going in terms of the time? Okay. Okay, so first 1 minute will be for this to finish to work. How much? So I have 15 minutes? Oh. Okay. Okay, let's see what. Okay, so we could. So another thing that one could study is ion channels. We finish most of what I want to tell you, but so in the case of voltage-activated ion channels, when the channel is closed, the ions don't pass through, but when you change the polarization on both sides, the channel open. So it's a general question, and you want to understand it again from a molecular or physical point of view. And it's very challenging. I mean, you could model by coarse-grain model the channel. You could do nice Langevin dynamics, but you also want to model the effect of the external voltage. And again, it's something which is very hard to do with atomistic simulation because you have this electrolyte, which are completely essential. And their equilibration take a lot of time. You also have the electrodes, which the channel community don't consider. As Chris Miller said, it's taboo to ask about the electrodes. We want to understand them. So you want to put all of them in the model. And we did develop models with grid that represent the electrolyte. We taught the system to reproduce all the known test cases like the By-Huckel, Gouy-Chapman, and so on. And then we use this model to study this potassium channel and to see the activation. So the first good news is that we could reproduce a landscape of opening and closing. And in more recent years, we were able to go to greater details to look on these barriers and to reproduce what is called the gating current or the gating charge. What happened is that when the protein moved between different conformational states, it moved charges. This moved the electrolyte charges, and you end up with charge on the electrodes, or overall you generate current at relatively short times, sometimes microsecond, so you want to model it. And what we learned to do, we realized eventually, is that since we know to calculate the time dependent of the motion of the protein, we know to calculate the response of the electrolyte to this motion. We could calculate the actual current as function of time, and we end up with current which reproduce the experimental results with slightly different interpretation of what happened in the protein. So this is ongoing type of study. We ask similar question of could we reproduce the voltage which is being generated on the electrode when a molecule like cytochrome C oxidase is activated. So there are a lot of nice questions that one could ask. And since I still have 3 more minutes, I will show you another exercise. Ada is not here, so this is for her. So if we have the ribosome, which structure was solved by these three people. It synthesized this peptide chain, which supposed to decide where to go. It could go to the membrane for membrane proteins or through the membrane. And there are really nice experiments that von Heine did, which look on the case where the peptide synthesis was stalled due to having unfortunate sequence that does not want to move forward. And then some of this peptide, when they start to enter through the translocon, were released from being stalled. So it's extremely nice story which go like this. You have peptide synthesis. You connect to the peptide a linker, so you control the length of it. And you try to see if the peptide is not released, the ribosome is still bound to the translocon. If the peptide is released, you could isolate the two guys and know that it was released. So you want to model here several very complex processes. One is the stalling of a peptide in the ribosome. The other is the rescue of the stalled peptide by the translocon. So you have to model the chemistry in the ribosome, which we and Johan Aquist have done for a long time. So we know to model this reaction by our QMMM approach. You also want to model the fact that for some sequences, the barrier for chemistry is higher in the ribosome than for other sequences. So we also model this. And then we try to model what happened to the peptide when it goes through the translocon. It's a long story. We found out that the last thing that this system will do is to try to move directly from the ribosome to the translocon, because then the barrier will be the sum of the barrier in the ribosome and the barrier in the translocon. So this picture explains it. And after all, we were able to simulate by Langevin dynamics the overall process for peptides which are being stalled and peptides which are being rescued. And we got this nice movie. Oh, sorry. So this is Langevin dynamic simulation with the right features. For example, the time of the peptide synthesis correspond to the experimental time. This peptide, after they bend a little, start to go into the translocon, and if they are of the right length, the force of going downhill in a later step in the translocon pull this part and release it from being stalled. So this is to show what type of questions one could ask with this type of coarse-grained multi-scale models. In all cases, you have to go back to the molecular physics to be sure that what you do is correct. And I think I will finish here. Essentially, these are different people who work on different projects, and thank you very much.
Okay. You beat the extra time. That's good. Thank you.
Yeah.
Any questions from the audience?
Okay. Okay.
Okay, right here. Yes. You need the microphone down front, please. Thank you.
Well, maybe you could say a little more about the coupling of the ATP hydrolysis to the movement, because in some of the models we've looked at, the ATP hydrolysis is primarily to release the back leg so you can start over again, and everything else occurs spontaneously by, and so I wonder if you want to comment on that.
Let's see.
In these motors.
My arguments are basically that in order to ask or formulate the question, it's very useful to have this type of landscape. Now, the place where the energy is released is actually when the phosphate goes to water. Doing ATP to ADP, which in water is 10 kcal per mole, does very little in the protein. It's essentially very funny. What happened in water drives the reaction. But the depth of this point reflect already the phosphate going to water. So my main argument is that you need the right energetics, but you also need the correct shape of the landscape in the configuration of these 3 subunits and the stock in order to have any tendency of rotation of this part. So you need the downhill energy. If you don't have downhill energy, you will have, yes, if you don't have downhill energy, you will not go in this direction, and then you will not have stock rotation.
Yeah. Okay. Plenty of time for discussion later.
Okay.
Think about your questions. Thank you very much again.
Okay.
Thank you. Wow.